Hi, my name is Željko. I am a teaching and research assistant at the School of Electrical Engineering, University of Belgrade. Welcome to the video explanation of the poster titled Composite Hydrogel Reservoirs for Controlled Delivery of Cationic Drugs. First, I want to explain the motivation for our research. Controlled drug delivery is important for drugs which are not suitable for conventional use through the oral delivery route. Such drugs can become degraded by metabolism, cause severe side effects and toxicity, or simply be less efficient if their delivery is not controlled. Cationic drug formulations are promising because they can enable passive delivery controlled by ion exchange, or active delivery by applying electric current, that is, via iontophoresis. Hydrogels are good drug reservoirs for hydrophilic drugs and highly biocompatible materials resembling soft tissues in the body. However, they commonly have poor mechanical properties. Addition of a hydrophobic polymer can be used to provide mechanical support and enable better control of drug release. Our idea was to produce a composite reservoir enabling controlled passive or active release of cationic drugs driven by ion exchange. Let us talk about the concept of ion exchange. Ion exchange is a charge-selective transport due to Donan equilibrium. It is enabled by the existence of fixed charge groups along the polymer chain. The illustration here corresponds more precisely to cation exchange, where fixed groups in the hydrogel network are negatively charged. Cations of the drug are exchanged with the cations of the medium, and the drug is released from the reservoir to the medium. This process is driven by the concentration gradient of the cationic drug between the reservoir and the surrounding medium. The process also depends on other parameters, such as pH, ionic strength of the medium, temperature, etc. What materials did we use for the fabrication of composite reservoirs? We first prepared the precursor solution by dissolving all functional components in a common solvent. Monomer and crosslinker were used to produce the polyacrylic acid hydrogel, while the polymer base will later become a hydrophobic matrix. Note that polymer bases are different for membranes and implants. Photoinitiator is required for photopolymerization under UV irradiation. With these materials, we perform the synthesis comprising two main steps. As we can see for membranes, the first step is UV irradiation of the solution cast on the glass plate, which leads to the formation of the gel. The irradiation was carried out under nitrogen purging. The formed gel was then quickly immersed in the water bath to finalize phase separation and solidification of the membrane. Similar synthesis concept was used to synthesize the implants. Now, the precursor solution was dispensed in the mold, constructed by sealing the Teflon ring at the top and bottom with a non-stick foil. The solution was then subjected to UV irradiation to form the gel in the mold. The gel was finally dropped from the mold into a PBS bath to finalize phase separation and solidification of the implant. We then investigated the different properties of the obtained composite reservoirs. We started with the characterization of chemical composition using FTIR-ATR technique. In the case of membranes, different wet surfaces have almost identical composition, indicating good symmetry. Bands corresponding to the formation of polyacrylic acid are evident in the spectrum, while the characteristic bands of polyether sulfone as the hydrophobic matrix also appear. We observed no significant shifts in the bands, which indicates mainly physical blending of separated phases. FTIR-ATR spectra of implants show similar features. Bands characteristic for polyacrylic acid hydrogel are present in the spectrum of wet implant, while the bands corresponding to the hydrophobic polymer matrix dominate in the spectrum of the dry implant. Absence of band shifts also indicates physical blending, as in the case of membranes. We also examined the microstructure of the membranes in implants. The membranes are quite porous and have an approximate thickness of 85 microns in the dry state. Hydrogel-rich particles of submicron size in the dry state are attached to the porous hydrophobic support. Microstructure is more complex in the case of implants, but shows no indication of porosity. In the bulk, implants contain densely packed hydrogel-rich particles of submicron size dispersed throughout the hydrophobic polymer matrix. Near the surface of the implant, hydrogel-rich particles form small scattered clusters embedded in the hydrophobic polymer matrix, which indicates lower hydrogel content. This structure results from the so-called skinning effect, common in membrane synthesis by immersion precipitation. The surface region with lower hydrogel content can act as a diffusion barrier for drug release. We investigated the kinetics of in vitro release from the membranes in implants using methylene blue as the model drug. Example of release kinetics for membranes is shown on the left. The kinetics is obviously pH dependent. At low pH, the carboxyl groups of the hydrogel are protonated. Membrane deswelling occurs and the release is faster than at pH 8. At pH 8, the electrostatic interaction with negatively charged deprotonated carboxyl groups slows down the release of drug cations. Release kinetics for disc-shaped implants is shown on the right. Release kinetics is significantly slower than for membranes, and predictable in vitro release under physiological conditions lasts for several weeks. Kinetics of passive release for membranes and implants is diffusion controlled, and the apparent diffusion coefficient is 1 order of magnitude lower for implants compared to membranes. Some additional tests have shown interesting properties of membranes and implants. We performed an experiment to demonstrate the possibility of active methylene blue release from membranes via iontophoresis. The experimental setup contained a membrane sample loaded with methylene blue, porous mechanical support, and graphite electrodes placed in the side-by-side cell filled with alkaline buffer solution. In this experiment, we achieved the zero-order release kinetics of methylene blue. Swelling kinetics of the implants was quite fast and even comparable with some superporous hydrogels. Equilibrium swelling degree in PBS was reached within 6 minutes. The swelling was reversible and the implant retained its geometry during the entire process. Described swelling behavior of the implant is suitable for clinical use since it can be stored in the dry state and rapidly rehydrated when necessary. Membranes can also be used for several applications other than drug delivery. They are suitable for the fabrication of nanoparticles, which can be either attached to the membrane surface or precipitated in the vicinity of the membrane. Hence, they can be used to produce membrane-supported catalysts or for the membrane-controlled synthesis of nanomaterials. Electrical conductivity and mechanical properties of the membranes also indicate the potential for the use of membranes as flexible electrodes. We can draw several conclusions from our research. Hydrophobic polymer matrix in the composite reservoir acts as an effective diffusion barrier, limiting cationic drug release. Adjustable and commonly slower drug release kinetics can be obtained by using the controlled skinning effect. Properties of composite hydrogels can be tailored during the synthesis by changing the formulation of the precursor solution, by changing the irradiation type, for example, by using gamma irradiation or electron beam, which eliminates the need for a photoinitiator, or by changing the composition of the non-solvent bath. The fabrication of composite reservoirs is simple, robust, and easy to scale up. Obtained materials are versatile and can be adapted for diverse applications. I would like to acknowledge the funding of our research and to thank our colleague George Veljovic for acquiring the SEM images shown in this video. If you are interested in the details of our research, check out our publications. The links are provided in the description below, where you can also find my contact information. Thank you for watching.